Good afternoon. Welcome to the Academia of Science for this special event. This is we call it Life Science Day because we are talking and then we have a distinguished scientist presenting their activity in that field. And particularly we have Professor Sylvia Daunert from the Miami University and Leonidas Bachas, which is the dean, or you said rector or chancellor of the Miami University. And we talk about possibility the collaboration between the two sides. And then, but more detail about the presentation of the Sylvia Daunert activity and Bachas, Leonidas will done by the chairman of the section. My just duty is just to introduce the president of the Academia, Luigi Bolondi, that for the welcome to the people here. Please.
Thank you, Aldo. Thank you for the presentation. I'm the president of this academy, and I am very proud to host in this beautiful place our famous colleagues Sylvia Daunert and Leonidas Bachas. We took the opportunity of personal visits of these two professors in Italy for inviting them to have a speech in this academy. And I thank you for the participation. You know that the academy is a very old institution of the city of Bologna. Its history is lasting from over 3 centuries. And this room is a room of the 16th century with the famous frescoes representing the history of Ulysses. And also the room on this side is representing the history of Ulysses. So we call this the Sala of Ulysses. So I'm very happy, again, I tell you, to listen to this presentation. And now it's my pleasure to give the talk to Professor Credi, who is the Prorector for the Research of the University of Bologna, for his welcome to the audience, please.
Thank you, Professor Bolondi. Good afternoon, everyone. I'm really pleased to be here and to welcome our distinguished guests, Professor Daunert and Professor Bachas, on behalf of the University of Bologna, on behalf of our rector, who's out of town today, but who is really keen to be here to testify also the close relationships between our university and the University of Miami, not only through Professor Roda, and thank you for making this possible, for organizing this afternoon, and also I'm grateful to the Accademia delle Scienze. I also have, as we discussed earlier, a personal relationship with your university through a very good friend and collaborator of mine, Francisco Remo, who also has a strong legacy with Bologna, as you know. So there are many reasons why it's particularly appropriate to host you today in this location, in this venue. I have to apologize because I will not be able to stay to hear your lecture, although that would be very interesting for me also because of my discipline. But I see 2 keywords that I would like to emphasize, which are very important for our university. We are very committed in investing on that, which are technology and translational. This is very important. This is the future of health. This is the future of medicine. And of course, by consequence, also the future of the related disciplines. So in Bologna, we have a very, say, fruitful activity on these topics, thanks to the collaboration between our departments of medicine and chemistry, engineering, physics, biology, and also thanks to the, say, strong synergies that we can make, and we are working actively also to improve that. The synergy that we can make with our university hospital. We have a large university hospital here, one of the largest and most, let's say, renowned hospitals in Northern Italy. So this is important for us to develop. And in a few years, we hope that we will have a new institute in the heart of the hospital, what we call the Biomedical Tower, where we aim at implementing translational research and biomedical research in collaboration between the university and the hospital. So I don't want to steal further time to the lecture. I hope to have the opportunity maybe to see you later after the lecture for a few minutes. And I was really pleased to meet you, and again, welcome to Bologna. Thank you.
Thank you very much. Now, before to start with the speech, with the lecture, there will be a short ceremony because Sylvia Daunert, she is a member of our society, but she didn't yet receive the diploma, and as a member, as a foreign member of the. So this is the reason why we dress her with the medal, with the president to deliver to Sylvia the diploma of the please, by the president.
As Professor Roda told us, Sylvia is now a member, non-resident of our academy. So I'm very proud to give her the diploma of the non-resident members of the society. Please look at the diploma. Take a picture, please. Take a picture.
No, you know, why not? We cannot stay. Adesso è ok, Presidente. Okay. Cheese.
Okay, get the picture.
It's an honor. It's an honor.
And now, Professor Cocco, Lucio, please come here that you are to chair the two sections. Okay, Professor? Professor?
Okay.
Good afternoon to everybody. You know, that's here we are in this marvelous room, the Ulix's room, and actually I should speak in Latin just to emphasize the importance of this. But you know, Latin was the language of science, the language of everything, of knowledge in the past. Right now is only English, is the only language that can unite all of us, both in science and also in life and the life science. It is a pleasure for me to chair this session, and actually it's also a pleasure because even though we have those two respected and very established scientists coming from US, but they have our same roots, Hispanic and Greek roots. So it's quite a privilege for me to say that. And you know, we're gonna listen a lot of things about new technology for translational sciences, as well as it's possible to build new strategies between the University of Miami and the University of Bologna, our alma mater Studiorum. And let me just introduce briefly, very briefly, because, you know, when you have to introduce foreign scientists or scientists everywhere, you know, we'll start, oh, he was born and then he got this, etc., etc. I think it's important to outline what they actually do and what is their field of interest. And mainly, I want to underline that Professor Sylvia Daunert, and she is a chair of biochemistry and molecular biology at the Middle School of Medicine and director of the Joy Macdonald Foundation Biomedical Nanotechnology Institute in the University of Miami, a very nice place, actually. I've been in Florida last summer. I was not far from Miami for a FASB meeting, and actually it's a wonderful place where you can live, even though there are too many mosquitoes sometimes. But you know, just apart from joking, what I want to underline about Professor Daunert, that she was honored with a very important recognition, the Hispanic Woman and Distinction Award. I think this is very important, and well as it's important what the award, the diploma that we gave her a few minutes ago. You know, it's important because on one side is my signature there, but you know, you must know that this is an academy that goes down until 1969, when the name was not the Academy of Science, but the Academy of Unquiet People. You know, it's absolutely unbelievable, unquiet. Some people that were thinking about what's going on in the world. And I want to underline the fact that she works on something very, very important, because her group genetically engineers living cells and proteins for environmental detection, molecular sensing, molecular diagnosis, post-of-care tests, biomarker identification, drug delivery. So that's really fantastic, because we're going to listen to something which is actually at the border of our knowledge at this moment. And I do believe that we will really enjoy, or we shall actually, to be real English, we shall really enjoy what you're going to talk tonight.
I hope.
Let me later on to introduce Dean Leonidas Bachas. You know, Spain and Greece. We are just in the middle, so that's perfect. This is a trilogy for what science can make. Please.
Grazie. It's a big honor for me to be here, and I'm really grateful to the Academy for inviting me. So I hope, and especially my friend and collaborator for so many years, Aldo, Professor Roda, I guess that he knows, we know each other from the time that I was starting my own independent research career. So that tells you, you know, that how far have we gone. So, you know, thank you again. And I'm going to share a little bit what we do in my lab, not all the projects, but to give you an idea. And I'm not going to go very deep into the projects, just to give you a flavor of the things that we do in the lab. So in general, one of the things that we always want to do is to address global health challenges that need solutions, all right? And there are many. We can go from agriculture to diseases to public health, like what happened a couple of years ago with the pandemic, right? So we need to have solutions at the tip of our hands. And for that, the idea is always to try to find enabling technologies that can actually do something, that you can make them useful and translate into a biomedical solution or an environmental solution. And for that, we always need to, borrow from disciplines, molecular biology, biochemistry, engineering, to develop bio-nanotechnology solutions, which is what we do in my laboratory. So if you look at our trajectory, the trajectory of my lab, and it's many years now. I started my independent lab in 1994. We were really one of the first labs that did some genetic engineering of proteins and cells and used them in biosensors and to develop technologies. And from there, we really started designing different kinds of proteins through the years and incorporating them into microfluidic devices. That was big in the late 90s and the beginning of the 2000s, and then it became commonplace. So we have been evolving with times, and one of the common things that you're going to see is that we really look at what are the needs at the time and try to apply whatever we know how to do in the lab to find solutions. So I'm going to give you a flavor of the things that we have been doing more recently, but with a perspective of what we did in the past. And I really want to talk about bioluminescence a little bit, and that is really how we connected with Professor Roda. He is a giant of bioluminescence. I'm not a giant whatsoever, but he is. And he opened up many avenues of using bioluminescence in analytical applications and in pharmaceutical applications, etc. And, bioluminescence is a phenomenon in nature that is beautiful. And even the US Postal Service in 2018 issued a whole booklet of stamps just with the organisms that bioluminesce. It was really special and very beautiful for all of us that really love bioluminescence. So bioluminescence, and Aldo has pioneered the field of really using bioluminescence in analytical applications, can be used for developing assays, imaging, etc. But there are ways and ways to do that. You can use the proteins that bioluminesce as such or you can do a little bit more fancy things and engineer them to use them in different applications. So that is what I'm going to be talking about right now. I'm going to talk about how we use bioluminescence to look into the microbiome. And the microbiome now is something that is very much in fashion. You have heard about, okay, what bacteria and microbes, what do they do in our gut? And I like to think about bacteria in our gut as movers, shakers, and barrier breakers, because they are continuously moving around. They really shake things. They fight with each other. They fight with the human host cells, and they even break the intestinal barriers and cause disease. So what happens with that and the microbiome? What is the challenge? The challenge is to understand what the role of the microbiome is, both when we are healthy and when we have a disease. And the solution is not simple, and it requires a team effort from many different kinds of individuals. And the little that I'm going to show you of what we do in microbiome today shows that. I mean, I work with world-renowned gastroenterologists, neuroscientists, OBGYNs, oncologists, fertility doctors, and of course a team of analytical and biochemists, analytical chemists and biochemists. So our solution focuses on looking at the microbiome, but at the molecular level, what happens with quorum sensing. And what the current microbiome analysis, how it's being done, is you must have heard about gene sequencing, C16S, metagenomics, metabolomics, transcriptomics, bacterial culture. Okay, all this is very important because it's going to tell you what kind of bacteria are present and the genes that are being expressed and proteins. But really, we don't understand at all what happens at the molecular level. How do these bacteria communicate and what do they do? So in that respect, we're looking at bacterial quorum sensing, which is how bacteria talk to each other. So it's analogous, let's say, as neurotransmitters in the human body, but quorum sensing molecules are small molecules, chemical molecules, that are being used by bacteria to talk to each other and to talk to the human host. And our idea is, okay, we need to learn what's happening at the molecular level, how pathways are being influenced, and why not to try to elucidate a fingerprint of these molecules in a given sample. If we know the fingerprint at a given time, then perhaps we can understand if these can be biomarkers of disease, or if we can use them for managing disease or screening for new drugs. So in that respect, again, I turn to bioluminescence, and what we have done is develop tools to be able to study the microbiome at the molecular level. And these tools are biosensors. Biosensors that are being constructed, designed by making cells bioluminesce in the presence of these quorum sensing molecules. So we can actually develop biosensors that will correlate the amount of that quorum sensing molecule with the bioluminescent signal being emitted. So we did quite a lot of molecular biology and genetic engineering, and we have been able to develop a whole host of different biosensors that detect different quorum sensing molecules, such as short chain homoserine lactones, long chain AHLs, autoinducer 2, DPO, and another one that is from Bacillus called PHRG. So now that we know and how to detect these molecules, we have the means to detect these molecules. And I have to say that we did pioneer this field. We were the first ones that were able to detect these molecules. Now we can start asking questions. What are the host microbiome interactions? So how do bacteria interact with the humans? Who are the players? can we detect these molecules in human samples, in blood, in the gastrointestinal system? What can we do? One thing that we know is, oops, let me go back. One thing that we know is that in that when the the gut is healthy, it has a certain bacterial composition, microbiome composition. However, when the gut is diseased, there are much less bacteria. The diversity is much less, and also the population of bacteria is lower. So there are two different things here happening: less number of bacteria and less diversity. So definitely there has to have some correlation with these signaling molecules, but at the time we didn't know what. So we started analyzing, and using our biosensors, we were able for the first time to detect these molecules in the gut of healthy individuals and people who had Crohn's disease. So our first publication was back in 2006, and we were the first ones to establish that quorum sensing molecules exist in the gut. And after that, we started looking at populations of patients with Crohn's disease and healthy individuals, and also looking when Crohn's disease patients were in remission or were under inflammatory state. And the idea was, can we use these quorum sensing molecules as biomarkers of disease? So say, for example, right now, to know if a patient has active disease, it's based either on symptoms or looking at inflammatory markers such as C-reactive protein, that is a general marker of inflammation, and then confirm with a colonoscopy, because otherwise you cannot confirm, or with an MRE. So if we could have a better biomarker than CRP, which is a general inflammatory biomarker, then perhaps we can manage the disease a little bit better. And that's where we took blood from patients and blood from controls, and we can see that there is quite a difference, very marked difference between the levels of AHLs, those quorum sensing molecules, the short ones and the long chain AHLs in patients with Crohn's versus controls. We also saw that patients with ulcerative colitis have marked differences in different quorum sensing molecules. So we are very excited about this. We just published a paper in the Clinical and Translational Gastroenterology, and that's where you see that we can merge the more basic science, like the development of a biosensor, with using it in a real life situation in a translational manner for detection of a development of biomarkers. We can also incorporate them in paper microfluidic platforms and use these biosensors at the bedside of the patient. So we did something similar with depression, clinical depression, and we actually see that because of the gut-brain axis, and this is a complex study to discuss in a few minutes that I have, but one thing to take as a message is that the microbiota in the gut really translate into finding these quorum sensing molecules altered when a patient is depressed. So there is a particular quorum sensing molecule, AI2, that when there is depression, either in animals or in humans, we also have done human samples, they are altered. And if you give an inhibitor of this AI2, the depression is being restored. So it's an interesting observation that we need to do further studies, but it's an interesting observation that the molecules in the gut really affect depression. And moving on to the reproductive tract, this study started because one of the fellows that I have in my laboratory, who is a fertility specialist, doctor specialist, he came to me one day and said, you know, all these women that we do fertility treatments and then we implant the embryo and it doesn't take, and we have no idea why. Do you think that maybe the microbiome plays a role? And I said, well, let's check it out. And so we started looking at cervical samples of women that had healthy pregnancies and women that didn't. And we certainly can see that there is a difference in quorum sensing molecules levels, two different ones, in women who had successful treatments and became pregnant and others that didn't. So the idea here is if we have a means that these women can be test themselves at home and be able to see if they have the right levels of the quorum sensing molecule that seem optimal for pregnancy, then perhaps we can facilitate a better outcome out of these fertility treatments. So more needs to be done, but this is an interesting project that we can use to predict IVF outcomes. And likewise, another interesting project that we're working on right now is looking at endometrial cancer. Endometrial cancer is one of the most malignant cancers in women. It's terrible. I mean, it's very lethal in women. And we are trying to see if there is a way to actually be able to look at and have biomarkers that can predict, number one, whether the cancer is there, and number two, if it is the degree, the grade of that cancer. And for that, we are working with gynecological oncologists, and we saw that there is a difference between high-grade cancer, endometrial cancer, and low-grade and versus controls. This is a project that is very much still going on. As you can see, we didn't have very much large numbers, about 23. So it hints that really there is a difference, but more studies are right now in progress. So we are excited about our progress towards understanding what these quorum sensing molecules do and how they are interacting in humans and the influence of the microbes in disease as well as in health. And with that, I want to change a little bit fields, and I want to go into the development of point of care tests and field tests. And if you look at history, even the Egyptians had developed tests for pregnancy. I mean, they were using the urine of women and barley grows to see if the women were pregnant. They were changing the colors. And then Robert Boyle developed the pH scale test, right, in paper. And then from there until 1943, they didn't come any kind of breakthrough until the dipstick diagnostics started, Helen and Al Free, which I happened to, have had the honor to meet when I was a graduate student. They really developed a series of dipstick diagnostics in Miles laboratories that later carried on, and still they are in use. And then not until the 70s, we didn't have a pregnancy test. And when you think about it, in the 70s, until then, there was no pregnancy test that was able to be at home. It seems amazing, but it's true. And, there has been an evolution of this point of care test where we have increased the microfluidics platforms being used. And ourselves, we work a lot with NASA, where we had developed technologies that are microfluidic centrifuge and other devices that NASA took into the space station and the shuttle. And from there on, things have been simplified. And in trying to address emerging challenges around the globe, we need to think that, yes, we need very simple solutions. We cannot have elaborate instruments because in many places, there is no refrigeration, there is no power, there is difficult access to location. people are not trained to use big instruments or difficult instruments, and there is a lack of hospitals and clinics. And this is not just like in the Amazon, okay? Because if you look at all these, all right, I tell you, this is Kentucky, where we used to live. So it happens all over the world, where there are remote locations. This is Haiti, where you need these kinds of technologies that are easy to use. And no one put it better than our friend Leonardo da Vinci. Simplicity is the ultimate sophistication, right? So we need to strive to have very simple type of solutions and devices. So in thinking about that, one of the things that our group has done is to incorporate, expand the genetic code of proteins by incorporating unnatural amino acids that can change the properties of the protein. So you can make a protein that was not capable of being stable at room temperature or at 37 degrees, which in a practical point, if you're going to have a sensor that works in an individual, it has to work at the temperature of humans and even at the temperature where someone has a fever, and it works at the temperature of the individual, the patient having a fever. So we can do that by engineering proteins, incorporating these unnatural amino acids that is going to make those proteins stable at hyperthermia and at physiological temperature. And for example, we develop a sensor for glucose by incorporating unnatural amino acids that allows them to work under those conditions, human conditions, and being constant monitoring of glucose. But these tricks, what I want to say is that we can play with these tricks and apply them to pretty much everything. Like we can apply them for precision agriculture, which is one of the new things that are happening nowadays with farmers. You wouldn't believe this, but you talk to farmers in North Dakota, and they actually want to have crops that they grow right there alone, and then they can monitor from their home. And this happens via satellites, right? They have sensors, you can put sensors in the field, and now those sensors are looking for the crops and are sending the information to the farmer, and from the station, then they can either add more nutrients or they can change the conditions whatsoever. So here are mostly this work that I'm showing in the next slide was done by one of our graduate students between Leonidas' group and mine, Harun Rashid. And I want to point out to here the detection of copper for better crops. Back in the past, and I say in the past because I published this in 1997, it seems like an eternity, but we use this kind of genetically engineered biosensors to detect arsenic in the environment, in the waters of Bangladesh, that they are very contaminated. Now, you can still use this type of sensors, but you can bring them to the 21st century, because 97 was still the 20th century, by incorporating other tricks. For example, we can use the green fluorescent protein and incorporate binding sites on that protein for copper. Copper is a nutrient for crops. So now we have developed a protein that we can incorporate as a sensor that it will bind to copper. And when it binds to copper, it emits light. And it will emit light in different intensities depending on how much copper is there. Then you can really put them in some agar discs and put them on the field. So now you're going towards that agriculture that is, that you can monitor, the farmer can monitor from far away, or can go with a cell phone or a device and be able to monitor the levels of copper in the field. So this is kinds of things that one can do to address the agriculture. So when we think about what happened in the past few years, right? Like the pandemic, the COVID pandemic and so on, and it caught us all out of sync. I mean, no one was prepared for that. But the good, if there is anything good that came out of that, was really the science. And so one of the things that brought to highlight was the need for tests for different infectious diseases, infectious pathogens. So in our lab, we have developed point of care tests of the kind that I said, that you don't need refrigeration, you don't need instruments, blah, 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 that you can visualize with your naked eye for a series of targets. For example, we started with the Zika virus, then HPV, HIV, et cetera, et cetera. And I'm gonna give you just like a little perspective on the types that we have developed. In this case, we developed a RPA type test, which is an RNA test for COVID-19. And in this particular case, we use this RPA principle, which is pretty much, to make it simple, like a PCR reaction, but without the need to change temperatures and use an instrument. You're doing it all in a little test tube with polymerases and enzymes that are able to replicate the RNA, and then we use a paper strip for the detection. So we incorporate nucleotides that can bind to antibodies, and then they can give you a reading and read. So it's not an antibody test. It's not the Abbott test that you use at home. It's much more sensitive because now you're detecting the virus, not it's not an antibody-based test. So much more sensitive. You can detect down to 60 copies of the virus RNA. And in our case, it had really great sensitivity and specificity. We work with the pathology labs of the university to develop this test. It's highly accurate and has no cross-reactivity with other viruses. So it was a really, really great test. Unfortunately, Abbott owns the reagents that we were using, and they blocked everything in terms of being able to distribute and commercialize it. But that's what, you know, Big Pharma does for you. If you're competing with their tests and they own the reagents for your different tests, then that doesn't happen. So we got a patent, a record in the US Patent Office. In 2 months, we got a patent for this test, but we couldn't commercialize it and use it because Abbott wouldn't allow us. That's one of those things. In that respect, you know, we have also developed a similar test for point of care for HPV, human papillomavirus, which is a humongous problem all over the world. And it has many different genotypes. It's a virus that is very challenging because it has so many genotypes that it's almost impossible. You may develop a test for one, and then another one comes, and you cannot really, that test doesn't work. So our trick was to look at the molecular biology, right? And try to design amplification primers try to find consensus sequences for the 14 most dangerous pathogenic types of HPV. And that's what we did. And we partner now with the WHO because our cancer center and the World Health Organization have an agreement and have designated our cancer center as the first one to collaborate for elimination of cervical cancer. So in our case, we developed a test for these 14, oops, okay, I don't want to show you the movie, 14 different genotypes of HPV. I have to say that HPV now, the only way to know, it's either they do like the inspection, the doctors and the pap smear test, or they send it for analysis in a laboratory, right? And then they do PCR and all these things. But there is no point of care test for HPV, none. And so we have developed this test, and you can see if there are 2 lines is positive, there is 1 line is negative. And our test compares extremely well. This is our test. with the gold standard laboratory tests that currently are being used. So the moral of the story is that we're doing clinical studies right now. We have done clinical studies, about 200 samples, but we continue collaborating and working with entities in Haiti and in Miami, where we can look at expanding the clinical studies. So continuing with this type of tests, bacterial vaginosis is one of the tests that really affects most women. And we partner with infectious diseases specialists that work in Africa and also in the Americas, and designed a new point of care test for bacterial vaginosis. And this is again a test where we did, I show very simplified things here, but it's not so simple because we had to actually go and engineer a protein, vaginalizing for detection of that protein, and then develop the LFA, the lateral flow assay for vaginolysis. And in addition to that, there are two things that you need to do. One is detect this vaginolysis, which is vaginolysis is a protein that the bacteria Gardnerella excretes in this disease. And also we could use a cell phone to look at the presence of what they call flu cells. So as I said, this is a disease that affects many women, and it's painful for them and can cause even infertility and can go without being treated. So the idea is to be able to have a point of care test that will allow for the doctor, wherever it is, in the middle of Haiti or in the middle of Africa, to have a simple tool that can detect this bacterial vaginosis and treat it in women. So we took a cell phone, and you can just get a little, it's like a sticker. It's really cool. A sticker that it's a lens that you put on your cell phone and acts like a microscope. And then you can look for these clue cells that if you put a dye, they will become purple and you can see them. And if they are healthy, they are pink. So it's a very easy solution for detection of this disease. And another very easy type of solution for a challenge that is very common is early detection of urinary tract infections. And that affects men, women, children, everybody, and especially the elderly, especially patients that are immunocompromised. It's a huge need. And the current tests for UTI or urinary tract infection are really very bad because this urine analysis that they do at the doctor's office, they look for leukocyte esterase test, which is very, very bad test, and same thing for the nitrate test. So really the gold standard is to take a little urine, sample of urine, and culture it. But that may take a few days, 2-3 days, and maybe you're missing treating the patient with the right antibiotic. So what we have done is, again, take advantage of our friends, the bacteria that are bioluminescent, in this case, Photobacterium legionati. It's a bacteria that emits light. But if there are other bacteria that are present and are consuming the oxygen that this Photobacterium needs to emit light, then the light starts to die. So we can actually develop a test, very simple test, that shows that when the light of the bioluminescence decreases, then there are other bacteria present and there is an infection. So here is a sample of patients that were positive for UTI, and these are the controls. And as you can see, clearly there is a marked difference. So we can use a very, very simple device that can be used in a physician's office to detect the UTI. Okay. Now, I talk about emerging pathogens. And you know, it took forever to develop the tests. But now we have a way to be able to develop them. We have a solution. And that is to actually be able to look into using in silico design, so bioinformatics mixed with artificial intelligence and machine learning to design what we call clamp peptides. And this is an idea that came out of discussions between Dr. Marcello Mascini, who is my collaborator. He's a professor at the Università degli Studi di Terramo, and he spends a lot of time in my lab in Miami, and he and I were talking because he's a specialist in bioinformatics. And then while talking, I came up with the idea, okay, why don't we design small peptides that are like a clamp, that they will clamp a target molecule, say a virus protein, like the spike protein, for example, from one side and then the other and grab it strongly. Why do I want to do that instead of using antibodies? Because number one, they are very small. So if you want to use them for therapies, they will not cause immunogenicity at all because they are not recognized by the immune system. And number two, if I want to design, if I want to use them for sensing or for targeting, also they are small. So they are much easier to use, and you can have really very good binding constructs. So, okay, I'm not going to go into a lot of detail how we do this, but it involves a lot of artificial intelligence design and docking and ways that we actually came up with some algorithms. My student Pepe Codina, a Spanish student that works in my lab, along with Marcello, you know, long distance mentoring between Marcello and Pepe and myself, we designed new ways to actually cut the time that it takes to design one of these molecules from, say, 1 month to 3 days, okay? So we can have screen libraries and libraries of compounds in just a couple of days and have a solution. So then we go and we do the experimental characterization of these small molecules, and then we do the antiviral evaluation, and I need to give a nod to my very, very dear friend, Joe Nichols, who is a professor at Houston Methodist at Cornell Vale and Houston Methodist, and she's an immunologist specialized in infectious diseases and characterizing and evaluating viruses. And so she does it in a very cool way. I'm gonna show you how. She actually is able to make 3D whole lung constructs. So she uses stem cells to actually build an artificial lung, a mini lung, human mini lung. And she also includes in there the immune system, macrophages, etc. And then she has a microfluidic device where she incorporates the 3D whole construct and seeds the virus. So in this case, she seeded, sprayed it with Omicron, okay, SARS-CoV-2 Omicron virus, and looked at what happens. And what you can see is that if here where you see the arrows and the red is cells from the construct that are dying because of the virus is killing them. Now, she took our clamp peptides and she sprayed them before putting the virus. And then, as you can see, there are only very few cells that die. So it really protects them. And if we look at the therapeutic efficacy, I just want to show you that there are marked different statistical differences in terms of pro-inflammatory cytokines that are being expressed. But more importantly, let me show you, this is more graphic, okay? This is what happens with the virus. It kills a lot of cells, whereas when you protect with the clamp peptides, there is almost non-killing. So you kill, the virus kills millions of cells, and when you protect with the clamp peptide, there are only 10,000 cells that die. So the mechanism, what is the mechanism? Without the clamp peptide, the spike protein of the virus binds to the ACE2 receptor and enters the cell. If we have our clamp peptide, the clamp peptide will bind to the virus, the spike protein, and prevents it from entering the cell. So it's a prevention. So our idea is we can use a mixture of these clamp peptides, spray them in the nose, and prevent the virus from entering. And when I say, COVID, it can be other viruses, obviously. So I don't know how I'm doing with time, but I don't know how I'm doing with time, but okay, let me talk a little bit about early detection in cancer diagnostics. If we look at prostate cancer, is the second cause of death of American men. There is not really a good cure for prostate cancer if it is very advanced. So we were set to think about how can we detect prostate cancer early. And we came up with this idea of looking at exosomes, using exosomes for cancer diagnosis. The person that championed this was Dr. Yuping Yang, who is an research assistant professor in my lab now. And we work with Alan Pollack, who's a specialist in prostate cancer, and his team, in addition to Daniel Bilbao, who is a cancer specialist too. So what are exosomes? Exosomes are extracellular vesicles that are being shed out of a cell. So if you have a cancer cell, these little vesicles are being shed out of the cancer cell. Here are the early exosomes, they come out. And in reality, if you look at them, they are like a mirror image of the cell that they come from. So they have the same type of RNA, DNA in them. They have pieces of DNA. They have all sorts of molecules that were in the original cell. So the advantage of exosomes is that because they travel, they are being secreted out of the cell, you can find them in blood. So what if we can use these exosomes to pinpoint that there is a cancer cell somewhere in the body, right? So that was the task. And the idea is that we can develop non-invasive diagnostics for early detection of cancer. So we developed this reagent that has a bioluminescent imaging probe, Gaussian luciferase, along with an antibody mimetic. Instead of using antibodies, we use DARPins, which are small antibody mimetics that bind to molecules that are expressed in the cancer cells and in the exosomes, okay? And then we put what we say a capture release handle for separating the exosomes that we fish out from other cells in a column. So basically what we do is that the exosomes have the same markers as the cell, and we demonstrate it here. And then we prepare these reagents and we pass them through a sample, we mix them, then we pass them through a column and we can separate these exosomes. And once we separate them, we can do all sorts of omics, you name it, metabolomics, transcriptomics, etc., to figure out what they have in there and be able to better diagnose the type of cancer that the patient has. So this is just to show that we can detect at very low levels, that we can use different markers so we don't miss any. So because if you have one marker and that exosome particularly, you don't catch that marker, if you have 3 different ones, you're gonna find them, okay? And so then we demonstrated it in animals, in animal mouse model of prostate cancer. And we do see that there is a marked difference between healthy animals and the ones that have prostate cancer. Now we can isolate the exosomes from the animal and do the molecular analysis that will give us clues on how to treat the patients. So we can use these reagents also, the same type of reagents, for imaging and early detection. And in this case, same type of constructs, and we can see that we can detect them down to very few cells in the body. Now we're not looking for exosomes, we're looking to find where the tumor is located in the body. So we're trying to find the cells that are cancerous in the body using these reagents. So we inject with these reagents, we can see we can detect them at very low levels. And if we put them, we inject them in animals, you can see that actually, let me see this. This movie doesn't work. Anyway, we can see that we can detect the cancerous cells in the body of the animal. What? It doesn't work here. Yeah. Okay. Sorry. Okay, so then you can see that we can find them in the animal. And what it is amazing is that you can actually image it with a simple cell phone. You can take a picture and see it. So for diagnostic purposes, it has quite a bit of value in imaging, but it's a very transient signal that doesn't last very long. So we thought, okay, maybe we can use a nanocarrier where we load a lot of these molecules that will allow us to image, and we can be able to see them for a longer time. So this is what we did. And so here we have a nanocarrier without the imaging luciferase reagent. And if we inject them into an animal, we cannot see anything. We don't see the tumor. However, when we have the reagent loaded into the nanocarrier, we can definitely see where the tumor is located, and we can see it in different kinds of tumors, subcutaneous, orthotopic, and for example, even in a renal capsule, all throughout the body. So it works quite well and for quite some time. Still after 3 hours and 6 hours, this is with a nanocarrier, and this is without nanocarrier. You see that we can see them for longer periods of time. So we're excited about this. Even with the naked eye, if we turn off the light, we can see how powerful the signal is, and we can see it in the animal body. Okay. I need to change gears now in terms of what I'm going to be talking about, and that is detection of volatile organic compounds. And this involves a huge team of people. We have 2 applications. One is in firefighters and the fire service, and the other one is in breath analysis in driving. And why am I interested in the firefighters? Well, we're interested because it's not always the fire that kills. In fact, the firefighters are at high risk of having cancer, and that is because they are being exposed to a lot of really carcinogenic compounds, in particular polycyclic aromatic hydrocarbons that they can get into the body through inhalation, through digestion, or even through the skin via deposition. So our collaborators at the University of Miami were the first one that documented how bad this problem is and created a registry in Florida that now has become, by federal law, a national registry. But it all started at our university. So what is our solution? Well, we created these silicone wristbands, like the ones that everybody wears, those silicone wristbands, and we modify them inside to actually absorb these polyaromatic hydrocarbons, and we give them to the firefighters. So the firefighters wear them whenever they go to one of their calls for fire or for a flooding, etc. So they wear them, and then they bring them back to us and we analyze them. And then we are able to create heat maps and see what they have been exposed to. And then we go back to them and we tell them exactly what they have been exposed to. So they are more cognizant of the level of the exposure that they had, and they can start with prevention methods. Another thing that we do is that we develop miniaturized sensors that can detect these molecules in real time. And we put them in little rovers These are like little toy rovers that are fit with our little sensors that are wireless and are sending information to us and to the firefighters. And we send them into a fire, close to the fire, enough for us to be able to monitor the levels. We also put them in the fire trucks, in fire stations, in bags and gear of the firefighters. And they monitor different molecules. So what do we accomplish with that? Well, actually, one of the things that we accomplished that we are very proud of is that the notion of these zones that are where the firefighters are, say that here is the fire, and the previous thought was, oh, maybe it's only here that is dangerous, the hot zone, but it's not true. All these compounds go all the way into the cool zone. And we were able to demonstrate that by using our sensors and our rover and also the wristbands. So what that led to is a change in the legislation also of Florida that has now made fire stations to have more and more filters to be able, HEPA filters to be able to contain the exposure to all these compounds, and also inform the firefighters that as soon as they are out of the fire, they need to go even further away. They cannot be stationed here because they are continuously being exposed to this carcinogen. So with regard to breath sensors, also another need, and this is in collaboration with BMW, the car company, they were very interested, they came to us, they are very interested in trying to prevent crashes, right? So people get drowsy, get tired when they drive. Is there a way that we can install some sensors in the car that will tell that the driver is not capable of driving because he's too tired? So this involves quite a lot of work. And first of all was to identify the breath profile, a fingerprint of a person when he's tired and is not tired. So we used medical residents for that because they have 24-hour shifts or 48-hour shifts. So we took a breath analysis before and after their shift, and we identified that they had different compounds there. So then we did we did GC chromatography of the breath, and we saw different biomarkers. And from there, we built sensors for those biomarkers that we can install in the car. So when the driver is driving, it exhales, right? Like the driver exhales. So we can monitor in the exhale of the driver, the breath, these molecules, and accordingly, we can know whether the driver is drowsy or not. So later on in the future, what our plan is that the car can automatically stop or send an alarm to the station and say, this individual is not fit for driving. So this is what we do with breath sensors. How am I doing with time? Like 5 minutes? Okay. So let me give you a taste of what we do with drug delivery. So you all know that with the COVID vaccine, now lipid nanoparticles and nanotechnology in terms of using nanocarriers came to life and showed how much nanotechnology is useful in applications. So in our case, we developed what they are called nanodrugs. So basically, it's a way to make drugs that already exist or molecules into small nanoparticles, so they are going to be able to be absorbed in the body much more efficiently than the aggregates, because the drugs that we normally intake are aggregates of molecules. And you need to have lots of them to actually be active. But if you have them separate in nanoparticles, they are much easily absorbed by the mucosa. and you can use less drug and have more effective outcomes. And one of the things that we're focusing on, one of the projects, is acute trauma. So when there is an acute traumatic event, such as brain injury by different causes, it can be a cardiac arrest, or it can be a heat stroke or a regular stroke, the current way of treatment is to try to prevent the cells, the neurons to die, right? So what it is like you target lowering the temperature of the body. So they put catheters, insert catheters into the patients to lower the temperature of the body. And if it is a brain, they put these kind of contraptions to try to lower the temperature, but it is very, very inefficient. It takes a long time to be able to lower the temperature, and then it may not be really that useful. So what our solution to that is actually to make nanodrugs of compounds that can create hypothermia, located hypothermia in the brain, by making nasal formulations of these nanodrugs that a first responder in an ambulance can deliver through the nose to the brain and cool the brain, okay? So we're working with neuroscientists on these projects, and we have been using vanilloids, which are natural compounds that have a cooling effect. They act upon the transient receptor protein potential vanilloid type 1 receptor, and by doing so, they can lower the temperature. So we demonstrated that we can make these nanodrugs by the method, it's called a reprecipitation method. So you take a lipophilic compound and slowly, you make it precipitate until you form the nanoparticles. You can see that we have nanoparticles here as imaged by TEM. And then we can take animals and not inject them, like spray them into the nostrils of the animals. And as you can see, we can lower the temperature to the therapeutic hypothermic levels that we need. Granted, this is not for long periods of time, but it's, if you compare first the brain and then the body drops, but first the brain temperature drops. So it's a very promising technology for treatment of therapeutic hypothermia, where you can get the results in minutes instead of hours like currently happens. Okay, I don't know if I have time. Okay, so another project that we're working on is on treating chronic wounds by using nanocarriers and delivering stem cells. So basically, we are targeting diabetic type wounds and open wounds. Like, for example, in hepatocellular carcinoma, when there is treatment by laser, by ablation, there are problems because the wounds in the liver are open. I'm sure that you know that, right? And so that compromises liver regeneration, and it causes loss of liver function. So one of my collaborators, Dr. Prasun Mohan, who is an interventional oncologist, came to me and said, hey, I know that you have this stem cell technology. Can we do something about it? Okay. So what we did is that we took nanocarriers, and in these nanocarriers, which are panamdendrimers, we put a targeting molecule, peptide, that finds the inflamed cells in the body, okay? And the nanocarrier will go to the locations where there is inflammation. Whenever there is a wound, there are inflamed tissue, there are these selecting molecules that are being recognized by receptors on the inflamed tissue. So we put them in the nanocarrier, and then we decorate stem cells with the nanocarrier, and we decorate the stem cells with the nanocarrier, and then we send them to the body until they get there, and they deliver the stem cells. So we did this first in mice, And you can see that even after 8 days, the wounds were healing when we were using mesenchymal stem cells, the nanocarrier and the targeting molecule. And if we just use stem cells without any kind of nanocarrier, the wound is open and it doesn't heal that well. So these were very promising results. And then we said, okay, let's try to look at the liver and use big animals for that, like pigs. In this case, we used a different targeting molecule, LFA1, which binds to ICAM, which is another receptor on inflamed cells. And we decorated the stem cells with the nanocarriers. So here is how the experiments were done. My friend performed a liver ablation, and you see the wounds here. This is the diameter of the wounds initially at day 1. And then we treat the animals with our stem cells. And after 30 days, the volume of the wounds had been reduced dramatically. You can see it here also after 30 days. And you can see also that that they regenerate the tissue. So it works, stem cells work, and having a nanocarrier really helps regenerate the liver, as we saw there. And this was just my last project. I don't know if I have time to show. Well, this is a different project where what we do is like we create biomaterials by using 3D printing technologies as a mold, and then we incorporate, we cast a polymer, chitosan polymer with a drag, and then we cross-link and we make these little sponges. And these little sponges, they contain SN32, which is a drug that is used in colon cancer. It's the metabolite of irinotecan, which is a very toxic compound. So the idea here was, can we do localized delivery of anti-cancer drugs? And not just put them where they need to go, but let them stay there and continuously deliver the drug after we put them after surgery. Why do we want to do that? Because then if there is any residual cells that are tumorigenic, then the drug will kill them. So this is what we did. We first studied the release of the drug from the implants, the chitosan implants, and through 3 weeks, they are continuously controlled delivery, which is beautiful, exactly what we wanted. And then we created tumors in animals, and we implanted these sponges into the animals. And we can see the survival of our animals, the ones that were implanted with the sponges that had the drug. They survived, totally survived. And the ones that they didn't have the controls, that either didn't have any treatment, or the ones that had a vehicle, which meant the sponges without the drug, they died. You can see this by ultrasound. We look at the volume of the tumor, and clearly the ones that had the drug, the sponges with the drug, had much, much reduced tumor size, whereas the other ones, they kept on increasing and died. And with that, I would like to really thank all the funding agencies that have, through the years, giving us funds to perform the work. Without money, you cannot do anything in science, unfortunately. It's very expensive. All this work is terribly expensive. So we are very grateful for all the agencies that funded our work. And also, this is a partial photo of my group. We have many more people, about 50 people in the lab, maybe more right now. So it's a very multidisciplinary group with chemists, engineers, physicians, etc., many clinicians that without them, that they come to us with the problems, obviously, we would not even imagine all the possibilities of using our technologies. So, you know, I I work so closely with my friends, the clinicians, because as I said, without them, we are totally ignorant on what we can do with the science. We do science for science, but they are the ones that come with the problems. So thank you all for listening and for being here with me today. Thank you.
Thank you, Dr. Sylvia, for this quite amazing and very exciting view of the possibility of technologies. You know, in my mind, as an old scientist, as an old researcher, just that technologies are fantastic.
Thank you.
The only problem is how scientists and mainly how physicians can use technology, because sometimes they are not using it in the very proper way. That's my personal feeling, not feeling of the Academy of Science, but I do believe that it's important to think about this issue. It's a very big issue, yes. Very big academic issue. So I do believe that we can, for questions, we can wait the second talk and then to have a general discussion at the very end of this day, of this Life Science Day afternoon, actually, I should say. So thank you again. And now it's my pleasure to invite here for the other talk, Professor Leonidas Bachas. And let me make a joke. It's fantastic that at this time we have two names of the Greek history, of the ancient Greek history. You know, Leonidas talking in the Ulysses room. This is something of really quite strange. It's a very good combination, you know. And Leonidas, okay, don't fight against the Persians, but just tell us how not to fight, but how to have relationship between Miami and Bologna. But before I give you the starting of your talk, I want just to address something about his interesting curriculum. And as you can see, he's Greek, he graduated in Athens, and he spent a lot of time in Kentucky, you know, nice place of United States. And he has been there professor at the Frank Derbyshire Professor of Chemistry in there. And it's important to say that at that point, he started to build up a group of young scientists. You know, he's not only a scientist, but he's a very good, very well recognized organizer of scientific groups. So I ask you just to tell us how we can join our forces to do something good in science, please.
Since we have people on the web, I mean, the Zoom, it's better if I speak for here because there is no microphone down there. So what I'm going to do today, I'm going to try to cover three general areas. I'm going to start by giving you some facts about the University of Miami. Then we're going to spend a little bit of time about some of the things that us that are more in the administration worry about. In other words, what's the future of the university as the world is changing, what we should be worrying about in terms of educating our students instead of in terms of doing research, and then finish off saying, but as we do all these things and as we worry about how we come together, how we create effective collaborations, and give some ideas about how we can do effective collaborations between institutions. So a few things about the University of Miami. University of Miami is, when we're talking about sustainability and resilience, is an institution that actually has been very resilient, because what you see up in the top is essentially the first year of the university. Some money was given to the university to be established. That's in 1926. And then the big hurricane, back then there were no naming them, hit Miami, and that's what was from the area where the university would have been. So the university had to rethink about themselves, how they're going to survive. They went to a building that was an old hotel, which you see in the picture below. And through the years became what we are today, where we research one university that is recognized for its research and for its education. We have as a symbol the U, okay, from university. And oops, go back. So why the U and why the colors of the U? It is actually the colors of the orange tree, because Florida has a lot of oranges. You have the orange, you have the green, and the Y in the middle is the white flower. So that's a symbol of the university. Our mascot, mascot is the ibis. The ibis bird is the first bird to come back after a hurricane, the last bird to leave as a hurricane comes. So it's a symbol again of resilience. And this is a symbol actually of a 5-star hurricane. So therefore we live in an area of the world that we have to worry about things like climate change, sea level rise, and how you are in a city that can have potential issues, and how do you recover from those issues, and how you prepare for those issues is important part of our research and teaching. A few characteristics. We have 3 campuses. We have a medical school, we have a law school, we have a marine school. In other words, we are a comprehensive university. We are, by the standards of the United States, a medium-sized university, 10,000 undergraduates, 6,000 graduate students from all over the world. 13% of our population is from outside the United States. And we are not a public university, we're a private university. It is a University of Miami, but only about 25% of the students are from Florida. It's a highly ranked university. In the United States, we rank the universities based on their strength. Recent universities, R1 universities are the top universities in the country. We have a global presence, and this is the size of research programs, 420 million per year. And essentially, in recent years, we have a focus on expanding STEM and the life sciences. Some of the institutes, we have a new concept that we started a few years back of how we build collaboration. The concept is life is not divided by majors. So therefore, even though we educate students as chemists or biologists or physicians or historians, that's not how life is. Life is a complex situation where everything is integrated. So have a philosophy of creating cross-cutting institutes that will bring people from different disciplines into kind of like an academy, and we call them the Frost Institute. So the first Frost Institute that was established on the area of chemistry and molecular science. You see, it's not an issue about chemistry, it's an issue about molecular science, bringing all the molecular scientists together. We have an institute on data science and computing. AI, computing, data science is very important nowadays. If there is one thing that all the students should know, it's data science nowadays, because all the decisions we're going to be making in the future will be coming through AI, but also will be influenced by how we understand ourselves, data, and the importance of statistics. And then the next big institute we're putting together, an Institute of Mathematical Sciences, that Miami is the city in the United States that's connected to more foreign capitals with straight airline flights than any other city in the United States. It's not airport in the United States. It's not JFK's. It's actually Miami. It's not Atlanta. It's Miami. It's a city that, if you think about where it's located, it's close to Europe, close to Africa, close to South America. So it becomes now a crossroads where a lot of scientists and a lot of mathematicians will come around this time, and actually a little bit earlier. February is the best time to come to Miami. It's great weather to get together in workshops and solve big problems. So we have funding from a private foundation to establish this institute in mathematics. Additional strengths, and I'm not going to go through all of them. I will concentrate on Resilience Academy. This is an area that we think is very, very important. I mean, we know that the world is going to be affected by things like pandemics, things like hurricanes for us, flooding, a strong wind here. So we are bringing together the best minds at the university through an academy. We're also having parallel effort on how change in climate may affect health, and what even a few changes in the degrees Celsius, even one degree Celsius, will change how vector diseases, for example, are moving, in our case, from the south up to Miami, and how that may affect the onset of diseases. And I think Sylvia mentioned dengue in one of her slides. That's one of the things that we worry as well. Bionium is a plug for my wife because she's the director, but it's not necessarily a plug for that. It's a major center where all equipment to create nanostructures and microstructures are present. So all the researchers from the area of Miami, but also the university, can use to develop nanotechnologies. And that is located in the Life Science and Technology Park, where essentially it's a tech transfer plant. It's also the place where entrepreneurs and faculty can have their companies listed and advanced by using the university facilities. The university students who can work part-time in the companies or full-time in advancing the research. So this is a technology transfer center. Some of the other centers that we have, we have a center in genomics, where we're looking at rare diseases or origin of diseases. You can imagine high-end sequencing and computing that's taking place there. We have a simulation hospital because we do have a nursing school. So when we try to train our nurses, those are trained with mommies, if you want, or with babies that aren't actually babies. So if you're going to make a mistake, you make it on somebody who is not human. So in the simulation hospital, you can have the patient, which is essentially not a human being, behave like a human being, and create symptoms, and then we are asking the students to see how they react. Parallel to that, because we do have a theater program, we have our theater students become model patients, if you want. So we train them how to pretend to be sick, and therefore when they meet the doctor now before, when they're being trained to be doctors, have the opportunity to work with situations that may not have expected to be happening. Okay, let's talk a little bit about what worries us. What worries us as educators and as administrators. Essentially, what is the future outlook of universities? And I say that because we live in an era that we have interconnectedness across the world, global interconnectedness. We saw that in many different ways. I mean, the pandemic is one of them. Within a few days, we have COVID being all over the world. And of course, what we have seen recently is an explosion of the ability of artificial intelligence to behave like human, have human-like behavior. So these are the kind of things that we are having to start thinking about as we try to think about how we educate our students for the future. The first quote actually is very appropriate where we are. This is from Clark Kerr, who was the chancellor of the University of California at Berkeley. He made the comment in 1982 and specifically said there are very few places, somewhere in the order of 85 in the world, that have not exactly changed since 1520. One of them, of course, is this university. And what essentially was trying to say is that the universities have actually been able to survive for many, many times across periods of times like world wars and pandemics and church changes and influences and so on. So there are enduring institutions. However, they have to adapt and evolve. So, for example, in the United States, we started with the colonial colleges, which were small colleges. Then we went to the big public universities, which were those established with the Land Grant Act. The Land Grant Act said that states could sell the land that belongs to them and the money collected establish universities to help with the agriculture and engineering principles back then of the states. And then later on, of course, we went to the Humboldt model, like many universities in Europe, of which are the research university that couple research and teaching. We also involved in terms of the size. I mean, we started with small colonial colleges that had a few hundred students, now to university like my university, which is having about 15,000 students, considered medium size, all the way to universities like our neighboring institution, Florida International University, which has about 60,000 students. And I just heard today that University of Bologna is even larger than that. So universities have really, really evolved. But they need to continue evolving because of the changes that happen. These are a couple of books, 3 books that appear in the last 3-4 years with some very interesting titles: Robot Proof, The New Education, Alternative Universities. So essentially, the question here is our curriculum enough to educate the challenges that our students are going to face in the future? In other words, I was teaching too traditionally for a very long time, where the world is moving faster, and should we change the way we educate our students as we move forward? And essentially, we're at the fork of the road. And I don't know how many of you know about the Alice in the Wonderland story, but when Alice arrived to this fork of the road, and she asked the Cesare, where should I go? Which path should I take? The cat says, well, depends where you want to go. So in other words, you need to have a plan. And as Alice says, but if I don't know where I want to go, essentially the answer back says, you may arrive there, but it may take much longer. So in other words, we need to have both strategies and ways to arrive to where we want to go. And the decision we're making cannot be postponed. We need to make the right decisions. I will tell you some of the trends of higher education in the United States. First one concerns cost. The education in the United States is tuition-based, and the students pay enough money for the universities to run if they're private. If they're public, there is a lower tuition, but the states provide some subsidy. And we have arrived to the point of what's called cost elasticity, how much we can increase the tuition to fund the university versus how much the parents will say that this is not worth paying. So cost is one of the parameters. There is some change in perceptions. I don't know how much of this exists in Europe. The change in perception is that I don't need a university education to find a job. And in recent years, and through political reasons that probably you have heard on this side of the Atlantic, there is more and more of people that are actually saying this kind of comment. I can find a good job without having to go to university. We see companies like Google hiring people and promoting that they will hire people that don't have a university degree. So the perception about the importance of an education is starting to change, and it's starting to change among parents. It doesn't help that Bill Gates never graduated from college. Mark Zuckerberg didn't graduate from college, so they always refer to some examples of people who were very successful and did not graduate from college. In terms of research, there is, I will not say you want crisis yet, but there is fewer jobs for doctorates who graduate from fields like literature and history if they want to go to an academic position. So there is a change in how jobs are being advertised and secured, depends on what discipline you are. So there is a time now to think about how doctors in different disciplines support the disciplines and whether we need to be making changes. For example, a historian shouldn't necessarily be working only at the university, maybe work at the museum. Do we train them? However, do we give them the expertise that's necessary to do these alternative careers? The other thing that is changing a lot is the cost to do research. I mean, I mentioned about the Frost Institute, just buying the electron microscopes that went in there, that was $20 to $25 million. The cost to being current when you do research is not just the amount of money you're going to pay for reagents, it's how you're current in equipment and how expensive those are. And those expenses are probably the same in this country as well in the United States. Market landscape, we are facing a demographic shift. The number of newborns in the United States started becoming smaller than in the past. Same thing in Europe. Some of this is backfilled through immigration. A little bit of that happened in Europe. But if you think about where we're going, you saw what's happening in France nowadays. We are going to have an older population expecting to have benefits because they have worked all their life, but a smaller tax base to pay for those that will pay for those benefits. At the same time, because the population is older, what we see in the United States, more probably you see that here, is a lot of a public policy and decision made by governments is more towards favoring the older populations like the younger one because the older populations vote. So there is that trend also that is happening in the marketplace. The other thing that's happening is micro-credentials. We see more and more of our students wanting credentials, and they don't just want a degree, they want to also know that I have now this coding credential. So we're seeing that trend in the United States. And lifelong learning, we don't educate students anymore for their first career, we'll educate them for their fourth and fifth and sixth. So in other words, we shouldn't just give them skills, we should actually train them to be successful and learn how to learn. I'm going to give a couple examples of trends. So what you see here is a very, very complicated graph, but I'm going to try to explain to you. It is a graph about when a technology was introduced and what percentage of the US households have adopted it. So let's see, for example, if you look at the last one here, this is where, I believe this is not tablet, this is, this is tablets. This is where iPad and the tablets were introduced, and this is how fast they were picked up. So what you look from this type of graphs that early on in the history of technology, the slopes of these lines were approximating here, where nowadays the slope of the lines are relatively sharp. So new technologies are being picked up very, very fast. I'll show you the same data in a different way. This is, for example, electricity. Electricity was invented in 1873 and it took 46 years for 25% of US households to have electricity. You can go to television. It took 26 years for the same issue, mobile phone 13, web 7. So things are moving much, much faster now. Why they're moving faster is because of the ability of microprocessors and electronics to do multiple calculations very, very fast. And this slide is only to show you through the years, the different processors and their ability to perform and how fast the slope of this line is. This is known as the Moore's law. For those of you that have heard about it, essentially says that the power of microprocessor, power of computing increases very, very fast. And what does that mean? Kurzweil, which was a futurist, said the following: if we plot the computations per second, if you're going to spend $1,000 through the years, it follows this asymptotic exponential curve. And then he put at the same time, what is the computing power of a mouse brain, of one human brain, and of all human brains at the same time? And he coined, no, he didn't coin himself, John von Neumann coined this term, the term singularity. Singularity is when computers will be as powerful, I mean, actually a network of computers around the world could be as powerful as all humans at the same time. And that's not that far, right? So if we follow the same pattern, which by the way is expensive because computing is energy consuming, so therefore at some point you have to put that into the equation, you're going to have something that John Newman back in 1957, 58 said, the ever accelerating progress of technology and changes the mode of human life gives the appearance of approaching some essential singularity in the history of the race, beyond which the human affairs as we know them will be different. But I come and think about it now. I mean, we are holding an iPhone. We're pretty much are in that stage where iPhone is now part of our being. You may see it as an extension of our brain, an extension of our hand. When are we going to arrive to a point where a computer may be able to make all decisions for us and may actually outperform us in the way we do our business? And how should we educate our students about the world that may be happening in 20 years out or 30 years out? What should they know? But maybe it's here already. By the way, you don't have access to this. It looked like it was blocked in your country after it was introduced. But let's talk a little about generative AI. This is the ChatGPT has generated a lot of news over the last 6 months, and ChatGPT is only one of about 6 or 7 different programs that are out there. Amazon has one, Google has another, Microsoft has another. It is a technology that actually can communicate with you in a way that it feels like a human is talking to you. It's a technology that if you tell them generate art in the form of this building or this ceiling, it will generate something that will not be the same as this, but similar to this, and you will not know that the computer generated. So what is it? Essentially, this company took information, at this time, this information a little bit old, it's about 4 years old, but essentially created the Library of Alexandria. You remember the Library of Alexandria existed, well, until about 300 BC, and had about 70% of the human knowledge at that time in terms of books. Well, imagine that this company, through scanning, because now all books pretty much have been scanned through a Google effort about 10 years ago, 15 years ago. The web is in front of you, so a lot of the knowledge is already in a database. So the company has created an algorithm through which you can ask simple questions. Last one question that somebody asked, yeah, I'm a writer, are you here to make my job? Here's the answer that was pulled back out from ChatGPT. Essentially sounds like human, behaving like human. Maybe we're not there yet, but we are close to a time period where we have to start thinking, what will happen if you give an exam, or if you ask somebody to write a paper, ChatGPT can write the paper for that person. Would we know who wrote it? would it mean that it's cheating if somebody used it? Well, we have to think about how we're going to be using this this type of approach as we are educating our students. In terms of global connectedness, these are things that you probably know. There is a shift in terms of where we find work. I went to the United States from Greece. The United States is hiring people in India and Ukraine to do computing. The worldwide distribution of knowledge is there. There is a global marketplace. We are outsourcing. But at the same time, we have to think ourselves, if we're going to work together, meaning if we're going to work around the world, how do we educate our students to be world citizens, to work in teams, to understand other cultures? Again, that's the kind of things that me as a dean, I'm thinking all the time. And what programs can I put together to do so? So how do we prepare our students for unpredictable futures? Maybe we cannot predict the future, but maybe we can give them the tools to be ready to face any future that may be coming. So I'm moving now to why, therefore, we're looking for collaborations and partnerships. And it is because we think that as we move forward, the best way to educate our students, the best way to do research is to collaborate, and we shouldn't just restrict ourselves to what we do in one location or in one country. We have an Office of Hemispheric and Global Affairs at the university. We have established already a network of 11 other universities in the hemisphere. Hemisphere means North and South America, where we're thinking about issues collectively, where we're thinking about how information can be shared across the institutions. This is one of the models. I think that there are some models in Europe that are similar to this, but the idea there is that students who have access to information from different institutions should be able to transfer knowledge from one institution to the other, and should be able to take courses in one institution and the other. Here are the partnerships that the University of Miami have in higher education. And as you can see, there are a couple, actually 3 different situations where we have partnership with Italy. We have several programs that send students to the American University in Rome for semesters abroad. And we do have interactions with your university here, which is described in this slide. This was established in 2014. A member of this academy, that's a faculty member at the University of Miami, established it. It is a dual degree program where a student will get a degree in engineering here and come to the United States and spend a couple of years and get a master's in engineering. And therefore they can have degrees in 2 countries that will make them employable in both countries. And several of our faculty have collaborations with the University of Bologna and joint publications. Some of them are with, as we heard earlier, with Professor Roda, but you can see some other ones here. One of them with, I believe, was the speaker that was earlier on here. So we do collaborate with the University of Bologna. We do have a partnership already. What are the things we can do? So one possibility is what I mentioned. We have a concept that is now quite popular in the United States, where students stay on and get a master's after they finish a bachelor's. So that's something that Europe was doing for a while. So we do have interactions with other universities where students can spend, get a degree from one country, and then as they transfer to one of our programs, the master programs, they get some credit towards that master program, which essentially will accelerate the time to get a degree. There are several options that we can do that. We do student exchanges. The student exchanges are different kinds. I mean, you heard some of them earlier. I mean, where a student from a country will come and do their final project in one of our laboratories. Several of those students are doing it from Europe. We have formal exchange programs where students will pay the tuition, let's say, of the University of Miami, but will come for a year or semester at the University of Bologna. And the opposite thing can happen, where a student doesn't see any expense because they pay the tuition in the country where they come from. We have exchanges of faculty. What I want to focus, however, is in these two different options, because I think these are real opportunities. So it doesn't matter what the title of the top is. They call it COIL, Collaborative Online International Learning. Sylvia and I have actually experimented with this approach in the past, and it goes as follows. Let's say that I teach, I'll give you the example of the courses that we did. So this was a course on sensors, biosensors. So we taught a course on biosensors. At the University of Miami, the course starts, let's say, in August and goes up in December. The University of Tarragona in Spain, another professor was teaching a course in biosensors, but in their case, they start, let's say, in October and go a little bit later. We couple the courses. We couple the courses by agreeing that we'll cover some general topics, but nothing more than that. Total freedom to the faculty member to teach their own courses. But what we did is we asked students from across the two continents to work together as teams to solve a problem. So in other words, we gave them a project and they had to work as teams. They had to figure out how they collaborate. They had to figure out how they come together over Zoom. Back then it was not Zoom, it was another program. And then at the end, they had to give a common presentation. So essentially, we show them how they can interact with another culture, understand how people work, and team science. So this is the one approach which we have tried and worked very well. The second one is these paired faculty exchanges, and I can tell you how this works. And again, I don't know if it works in the current structures, but essentially says two people want to collaborate over a period of a year, two faculty. I will say that, let's say, my faculty member will come to University of Bologna for the first semester. I will allow them to do so by paying their salary and not expecting any teaching. The University of Bologna, we may assign them to teach something. Then the second semester, the University of Bologna person will come to the University of Miami, and that person will teach a course at the University of Miami. And the faculty member of the University of Miami will teach a course at the University of Miami. So if the teaching responsibilities for a faculty member is 2 courses a year, I have gotten 2 courses as an administrator, so therefore I'm happy, but hasn't cost anybody anything. So this is what I see are these paired faculty exchanges, which again can work as a way that people can come together and work together for a period of a year, spending half a year on one location and the other in the other. And I will stop here, leaving you with this slide to be watching without talking about it, and because I think it's time for questions. Thank you.
Thank you very much, Leonidas, for your very clear and encouraging way to how to start and improve the still existing collaboration. So now I think we have time for questions or curiosity or things to ask to our speakers tonight. Please. Questions? From the audience, Professor Roda, you are the one, you are in charge for asking something. No question at all. Now, I would just,
yeah.
Oh, yes, on you go, Aldo, please.
What is the barrier between technology, science, and business? Because, we are doing all this stuff at the university or part to do with the private company, with the, we call it mission, with the startup, or how is, what do you think? How you transfer to the real world? This is a question that involves both the scientists and the management of the university.
Okay, so as a scientist, like we have a really very active office of innovation and technology transfer. So, you know, there is not a problem to convince the university to patent your research, as long as you demonstrate that it is innovative, and then they bring in the lawyers, the attorneys, and they do searches, and they are convinced, okay, it's a novel idea, we're gonna patent it. But then the bottleneck is you get a patent, right? The university has invested a lot of money, but the key is to be able to find companies that are ready to either license the patents, or, you know, if you're a faculty member and you have your education mission and research, to start up a company and devote time to the company is very time consuming. So you have to be very realistic about that. I mean, at least in the United States, it's very hard to be able to do both. Either you need to think about, okay, maybe someone from your lab can take on and start up a company with the technology. But if you want to remain being a professor full-time, it's very hard. It's very, very hard. So that is very challenging. Licensing, it's a little bit better, but the problem with universities is that they don't have a very good infrastructure to match the innovation with companies. So that, I think, is where a lot of the technology is being lost in the part of the technology transfer to the industry. That is much more challenging because companies want a product. And when you do research at the university, you have done the part of demonstrating that it works, but then translating into a product takes much more than whatever you have done at the university. And investors don't want to fund that. Investors want to fund when it is already ready for a product. So early investment is still a very big challenge in the United States. I don't know how it is in Europe, but in the United States, early investment is a big challenge. That's where the challenge is nowadays, to translate the science into real things. So.
Any question? Come on, please.
I have curiosity regarding the last point that the Dean made regarding the transfer of professors from Miami to other university and doing the teaching in the Austin University. Did you already start this process, for example, with some Italian university in Rome or in Sweden? Okay.
So it requires that the people come together and they are willing to do this. And for me, essentially means that I have to be flexible in terms of the time this semester. Meaning somebody may come and teach at the University of Miami, but you may need to be earlier. I need to have somebody else fill in.
Sure.
But it did work okay. But we tried only to have a second.
And I guess you need also flexibility about the curriculum, like the content of the course. Thank you. That's very interesting and good idea, I think.
Because that's the whole slide.
I have just a very, very short comment, not a question. I believe that I am the only clinician in the audience. I don't know if there are other medical doctors with a clinical practice. So I am the only one. And I have to tell you, very distinguished, still practicing medicine. And I have to tell you that I've been very, very impressed by the very great amount of possibilities of applications of
the technologies you have illustrated. And I am sure, and I regret that the clinicians are not aware of these possibilities, the majority of the clinician. The clinician in your university are very lucky to have a strict collaboration. Probably also in Bologna, we can develop such collaboration with these technologies, but we still have many problems in making this translational research, and we lose many possibilities. But all the application you told about from the gut microbiome to the depression, to cancer diagnostics, to viral infection, there is probably all kinds of pathologies that can be investigated with these techniques. And fortunately, your lecture will remain in the site of the Academy of Sciences, and I will stimulate my colleagues of the medical faculty to look at the conference, because I think that they also will be impressed, and but they will have the possibility to listen to what you said.
I cannot take full credit for any of this, because actually what happened was when I joined the University of Miami in 2010, I was the first woman chair of a basic science department in a medical school where the clinical departments were dominated, and all the chairs of the clinical departments were males. They were very excellent colleagues, and they all welcomed me very much. So through meeting them, you know, and telling them a little bit of what I was doing science-wise, they invited me to their departments to give ground rounds, lectures. And there I got to meet a lot of clinicians, and they saw the possibilities, and they were coming to me like, they would come to me, hey, Sylvia, I have this problem. I mean, do you think we can find a solution? And that's how I started really working with clinicians. And every single project I showed today involves a team of clinicians, PhDs in different disciplines, and some of them engineers as well, and pharmacists. So it is, you know, through personal connections, but you have to be open-minded, okay? You have to be open-minded and willing to invest your time and resources. And sometimes, you know, the first time when you start doing this research, there is no money, right? You don't have a grant for that. But you get the data, the preliminary data, and then you can apply for grants from the NIH or the Department of Defense or whatever. But it is the willingness to collaborate. And, you know, the University of Miami, I would say that the best thing that we have is easy collaborations, because my department and Christiana has been in my labs, and Aldo has visited. We are in the medical campus, but we are all very close located. The research part, the research is close to all the hospitals. So, you know, my clinician collaborators can stop by the lab. Some of them have. I give them an office in my labs, in my we can actually work together. So it's the only way to start building it and opening the mind is through personal connections, and you willing to go give talks and tell them about potential technologies. And then you open up their mind and they like, oh, maybe I can use this for that. And then they come and they say, hey. So it's very enjoyable because, you know, I love science and I love developing technologies, but if they don't go anywhere, then, you know, it's kind of a little bit of frustration, right? Like it doesn't go anywhere. But if you see that actually they have a good application, it's very, very nice. So thank you for that. Thank you for the comment.
May I ask just one simple question? Do you still have, or do you have a good MD, PhD program in the medical school.
Yes, we do.
Because, and you know why? That's my question, because that's the point. We don't have in Italy, we are fighting, you know, the so-called preclinical with the clinical part and things. So when I said many years ago, we should start with an MD, PhD program, you know, the answer was, you're a stupid guy. But I do believe that it's the only way to really mix the basic science with clinical.
Yes, yes.
Clinical
definitely.
science. That's the only way. And
Definitely.
you are, you know, in the United States, you have many problems. Sometimes, you know, I've been scientifically British educated, so still is a colony, you know, America, but in terms of science. But you have this great point in the medical field to have the MD, PhD program,
Yes.
which really makes the difference. And
Makes
we
the
are
difference.
poor people that we are just saying, okay, but Italian medical school is the best in the world. You know, theoretically could be, but not in practical. That's my feeling, but you know.
No, the MD, PhD
It's not
program.
what all the people in the academy believe.
No, and you're going to like this. There is not only the MD, PhD programs, but now they also, there are, for example, our cancer center and many of the departments. So I mentioned that I have 3 fellows from OBGYN, okay, and gynecologic oncology in my lab. Why? Because they have fellowships for MDs to do research for 2 years. On the bench. I train them. I mean, like my people train them in the lab to pipette, because they never have pipette in the lab, right, in their life. So they learn from the very beginning, and they are so enthusiastic. So we train them actually to do research, but it's fellowships that the university has for MDs to be trained in research. That's very important. Very important. It's a gift, yes. And they add so much to my lab too, because then, you know, those clinicians teach my PhDs, you know, about medicine. It's very important to cross-pollinate. Anyway, excellent discussion. Thank you.
So as a chair of the session, I thank speakers and all of you.